let's start here. Okay, so then for water, we have a polar covalent bond between oxygen and two molecules of, two atoms of hydrogen. So in this case, you will have these electrons shared by this oxygen and this hydrogen. And then in here is the same. You have these electrons shared between this hydrogen and this oxygen. And because this oxygen molecule is larger than the hydrogen molecule, then these electrons are pulled more towards this oxygen because it has greater number of protons, which has the positive charge that attracts the negatively charged electrons. So because of that, you create this delta negative charge in here and a delta positive charge in here. So delta negative or minus and delta plus. So now let's look at hydrogen bonds. So hydrogen bonds are made of weak attraction between hydrogen and one molecule. So these hydrogen bonds are very weak in comparison to the covalent bonds, and these affect the formation of molecules in water because you'll have these charges in this hydrogen, which is positive, and this other one, which is positive, leaving it free to interact with the oxygen from another molecule of water. And that can create a bridge. And this bridge, it will form cohesion. Cohesion is basically a property where molecules can come close together. And that will increase the surface tension, and then this will prevent the water from rupturing the bonds. Although these are weak attractions of hydrogen bonds, however, when they're multiple, they create these strong structures, including the surface tension that exists in water, that helps for some small organisms to be able to walk in water. And then in terms of our body, water, it makes more than any other molecule in our body. So for females, we have 50% of body weight made out of water. In males, it's 60%. And this is important to several functions, and it serves as a solvent. So many molecules dissolve in water because we have water, and many chemical reactions take place. So here is an example then of these hydrogen bonds in a molecule of water, or in water, sorry. So here we have molecule of water, another one, another one, another one, another one, another one. So again, you have positive charges free in here, in these hydrogen molecules. And then these oxygens have negative charges. So you will form a bridge between this hydrogen, which has positive charge, and this oxygen that has a negative charge. And that forms a lattice. So these entire molecules of water, they come together, they form cohesion, and that helps to create surface tension, which forms, helps to form droplets. In this case, for instance, can be blood, and plasma in blood has a lot of percentage of water. So how chemical reactions happens? Well, it's not only having molecules or atoms, you have to have them nearby. And then this physical closeness helps them to be able to collide when they are in random motion. Or they can get attracted by an electrical charge. It's like someone who wants to make friends. Well, if the person is isolated, he cannot make friends, he cannot make chemical bonds. So it has to go to a place where there's people with particular interests that the person might like, and then you start chatting with the person, and then you start forming friendships. So you have to have this property of having physical closeness. And then in chemical reactions, you typically have enzymes that helps to speed up these chemical reactions and then can join the molecules, creating a chemical bond. Well, maybe you go to the right place where there's a lot of people with interests that you like, but you're a shy person and you don't like to talk as much. Well, still you won't be able to make a lot of friends, or maybe you won't. But what if you have a friend who is more chatty than you, and then it starts talking with other people and introduce you to those people? So that's like a catalyst, increases the chances of you making friendships. So that's an enzyme. And then the enzymes themselves, they don't have to change or deplete. And then in chemical reactions, products of the chemical reaction and energy are released. And enzymes reduces this activation energy for producing this chemical reaction. And then you have to have the right temperature, the right concentration of these reactants, and as well, you have to have a particular concentration of hydrogen ions, which is the pH. So pH is a scale, or is a measurement. So the concentration of hydrogen ions in pure water is 7. And anything above this, it will be basic or alkaline. So this is neutral. So basic or alkaline pH will be greater than or equal to 7.1. For the opposite, when you have lower than 7, so that will be acidic pH. So this will be let's put it this way. So it will be lesser than, that's the symbol for lesser than or equal to 6.9. So then temperature, concentration of the reactants, pH affects the chemical reaction, and of course, the presence of enzymes. Now, the form of energy that we can have stored energy or potential energy. So that will be the energy that can be used. And then we have kinetic energy, which is energy in motion when someone is doing work. And then both of them are interconvertible. So kinetic energy can be converted into potential and potential into kinetic. It's like when you recharge your batteries and then you use it. So when you plug your charger and then you put your battery in your charger, you are transferring electricity into your batteries. So that transfer of electricity coming from the electrical outlet, that's kinetic energy. And then as it's getting stored in your battery, that will be potential. It will be converted back into kinetic when you put in your device, and then you use that because you allow the flow of electrons. In our body, we have a type of energy, it's called electrical energy. That is the type of energy that basically is used by our neurons to transfer impulses. And then we have the chemical energy stored in molecules like lipids, like ATP, adenosine triphosphate. And this is a type of energy that will be released when we use these lipids or this ATP. And then we have mechanical energy, that it is produced when we contract our muscles, and that moves our joints, so that we can carry objects or can move from one place into another. And then in our body, we need enzymes to lower that activation energy so that these chemical reactions take place. And that makes our metabolism more efficient. So here's an illustration showing you what happens with this energy. So in order to roll a ball down the hill, and if you are in the bottom of the hill, you have to roll the ball towards the hill, at the top of the hill, and that will require for you to spend some energy. Once you are here, and you just push the ball itself will roll down, and that it is basically kinetic energy. Now, how would you reduce the amount of energy that is required to roll the ball? Well, in this case, of course, it's going down to a lower hill, which in this case, I mean, if you have one nearby, you can do that. But instead of going to a taller hill, however, in actual chemical reactions, what happens is like you can have the chemical reaction going, like if it was going into this tall hill, and then you still have the chemical reaction. However, that chemical reaction would take longer than when you use an enzyme. So here is the illustration as if someone was pushing the ball towards this tall hill versus someone pushing the ball towards this short hill. So this will be the work or the chemical reaction with an enzyme, and this will be without an enzyme. So the activation energy, which is basically rolling the ball up the hill, it will be reduced by the presence of enzymes. Now, in chemistry, you have chemical notation that you use for writing down your chemical equations. And The reactants typically are put on the left of these chemical equations, and then you put an arrow pointing where that chemical reaction is going. So in this case, we have a synthesis or an anabolic reaction that is basically having reactants A and B, or A plus B, that are going to form a chemical bond. So they were first separated, A and B, and now with this chemical reaction, which is called synthesis or anabolic reaction, you create a bond between A and B. So that would be AB. In the composition reaction is the opposite of synthesis. So you have catabolism. You break down the bonds between A and B, and then you have the products. So the products are on the right, reactants on the left, and the arrow tells you where the reaction is taking place. So in this case will be A and B, you break down the bond, and then you create A plus B. You can also have chemical reactions in which you exchange or transfer molecules. So in this case, you have A and B plus C and D, and you can have the following products: A and C plus B and D, which are different than this, or A and D and B and C. And you can have also reversibly transfer reactions in which these products can give you these reactants, or these reactants give you the product. So it can change depending on where the name can change based on where the chemical reaction is going. Like these can be reactants, can give you these products, or this can be the products, the reactants and give you these products. So in anabolic reactions, you take monomers. Monomers are single units. And then you join them, and then you can create a polymer. So this enzyme grab these substrates or reactants, put them in a place of the enzyme that is called the active site, and then it changes its shape to bring these substrates together. It creates a chemical bond, and they undergo something that is called the transition state, in which they will be converted into products, and once you have a product, you release it, and then the enzyme returns to its original shape. So that's anabolic reaction. So examples of enzymes deficiencies. So we have, for instance, the lack of an enzyme that is called lactase that breaks down the carbohydrates that we have in milk. which will be lactose. It breaks down lactose into glucose and galactose. And then if someone doesn't have this enzyme, the person will have lactose intolerance. That's a simple form of an enzyme deficiency. Another one is TASAGE deficiency, in which there is an enzyme that is missing that is called hexoaminidase. And this one affects the development of neural tissue. We have another one, another deficiency. We have the lack of adenosine deaminase in severe combined immunodeficiency. Someone who has severe combined immunodeficiency, the person will have a lot of infections that the person can die from. A deficiency for the conversion of one amino acid into another one is phenylketonuria. Is a deficiency of phenylalanine hydroxylase. People who born without this phenylalanine hydroxylase cannot ingest phenylalanine in their diet, because that will lead to accumulation of byproducts that will damage the brain. Okay, so now let's talk about inorganic compounds. So inorganic compounds are compounds that do not have carbon and hydrogen bonded. Organic compounds have carbon and hydrogen, while inorganic compounds, they do not. So examples of these important compounds in our body will be water. Can make up to 60% of the human body mass, and then water will be the universal solvent. And we have other examples of inorganic compounds like hydrogen ions, hydrochloric acid, carbonic acid, so HCl is H2CO3 is carbonic acid, and HCO3 is bicarbonate. So all of those are acids. An acid, it's a molecule that can donate hydrogen ions. And a base, it's a molecule that can accept hydrogen ions. So bicarbonate, it's a base. So this is a base. And these two, of course, are acids. So this is a beaker, and you have here pure water. Pure water has equal numbers of hydrogen and hydroxyl groups. Hydroxyl is OH minus, and hydrogen is H plus. Now, when you add an acid or a molecule that donates hydrogen ions, like for instance, hydrochloric acid, if you add hydrochloric acid here, that acid will dissociate into chloride and hydrogen. And the concentration of hydrogen ions is going to be greater. So this solution is going to be acidic. It will have a pH lesser than 7, so 6.9 and below. An acidic solution, like for instance, food that is acidic, like lime juice, it has high concentration of hydrogen ions. And then if you have a beaker with pure water, and if you add a base, like sodium bicarbonate, this sodium bicarbonate is going to dissociate in water, and this is going to bind free hydrogen molecules, forming carbonic acid. And that will decrease the hydrogen concentration in the solution. So in this case, the pH is going to be basic. So it's going to be greater than 7. So it can be 7.1 and above. While in here will be 6.9 and below. Part of our metabolism generates a lot of acids, including lactic acid. So we have mechanisms in our body that help us to get rid of that excess of acid. Like for instance, every time that you do exercise, when you generate lactic acid, you start breathing more. Because when you breathe more, you get rid of CO2, which is one of these molecules that it helps us to basically get rid of this excess of hydrogen when it combines with water, forming carbonic acid. So properties of water: absorbs heat, carries the heat, and it changes from liquid to a gas. Like when we sweat, we reduce our body temperature. It helps to cushion and protect body structures. It helps to lubricate and reduce friction, and is the solvent. So anytime that we ingest a salt, that salt, it will be broken down into individual components because we have water. And then because water is polar, it has a positive and negative charge, it will be able to help us to separate molecules into two categories. The molecules that can interact with water, which are called hydrophilic, and the ones that do not like to interact with water, which will be hydrophobic. And then pure water has a pH of 7, and in this case you have equal amount of hydroxyl groups, or OH minus, and hydrogen ions, which will be H plus. So this is basically how water is formed when you add hydroxyl groups and hydrogen ions. So then ionic compounds, or the ones that can donate electrons or accept electrons, are hydrophilic. One example, sodium chloride. So when you add salt into pure water or water itself, this sodium is going to be hydrated, and then this chloride is also going to be hydrated. So this will have a positive charge, so it's going to be a cation, and it can interact with oxygen. So see how these red spheres that represent the oxygen surrounds this sodium. Why? Because this has a positive charge and the oxygens have a negative charge. And then chloride, which is going to have a negative charge, it will become an anion. It's going to attract the hydrogen portion of the water, because the hydrogens have positive charge while this one has a negative charge. And that's how you separate them. So you form out of this compound where you have sodium atoms and chlorine atoms together, you separate them, and then you form molecules that now have a charge, which will be a cation, a sodium ion, and then an anion, which will be a chloride ion. And these ones can travel in the bloodstream, reach electrically excitable cells like muscle cells or nerve cells, and they can cross the plasma membrane and help to have our muscle cells and our nerve cells generate action potentials, which are important for the function of the cells. And now we have here an example of a polar hydrophilic compound, which is water, of course. So in this case, we have carbon monoxide. Carbon monoxide has a positive charge and a negative charge. So since this is a polar, this molecule, it will interact with water molecules. In the case of nonpolar hydrophobic compound in water, so you might have a molecule like methane, in which you have a carbon and then you have 4 hydrogen molecules, is CH4. This molecule does not have positive or negative charges. So if you put it in a jar, sealed jar with water, these molecules will remain on top while water will be in the bottom. There will not be any chemical attraction between them. Or like oil and water. That's an example of another nonpolar compound. So in this case, both of them are polar, water and carbon monoxide. And in here, this one is nonpolar and this one is polar because it has charges. So here is an example of the scale of the pH. It ranges from 0 to 14. And the lower the number is, from 7 and below, the more acidic the molecule or the solution is going to be. Like, for instance, our stomach acid is very acidic. Then vinegar, lemon juice are very acidic. Tomatoes, coffee, milk are acidic. And then here, in this left column, it represents the concentration of hydrogen ions. So it's high, and then the concentration of base is low. And then the greater the number above 7, the more basic the solution it is. That means that you have less hydrogen ions, but you have more base. Example will be bleach, ammonia, baking soda, and blood, which are a little bit basic. In our plasma, the pH is slightly basic, between 7.35 to 7.45. And then another physiological importance is that many enzymes work at a particular pH. Like we have digestive enzymes that are working at a pH that is basic, and others that works at pH that is acidic. And they are located in areas where we have acidic fluids and areas where we have basic fluids. And then imbalances can cause acidosis or alkalosis, disrupting homeostasis. So this is the pH of plasma, 7.35 to 7.45. When you have lesser than or equal, well, actually it will be less than 7.35, that leads to acidosis. When you have greater than 7.45, that leads to alkalosis. So in this case, the concentration of hydrogen ions, concentration is in brackets, is increased. And then in here, the concentration of hydrogen ions will be decreased. So organic compounds are molecules that has carbon and hydrogen linked, and examples will be carbohydrates. Carbohydrates have monomers that are called monosaccharides. And then these monomers can form chemical bonds, and then you form polysaccharides. And the general formula of carbohydrates is CN, H2N, and ON. So C and O will be the same number, and this will be twice the number of carbon and oxygens that you have. Then proteins. I'm going to skip lipids because I'm going to give you an example of monomers and polymers. So this will be made out of monomers that we call amino acids. And the amino acids that we have to make proteins are 20 different amino acids that in different combinations form different proteins. And these proteins, they have a diverse function in our body. So, same for DNA, RNA. They are made of monomers that we call nucleotides. And the nucleotides have a nitrogenous base, sugars, and a phosphate group. So, this is an individual marker. This is a monomer. If I attach another marker here, if I join them chemically, this is a dimer, right? It's made of 2 units, 2 monomers. And I add another one, trimer, tetramer, and I keep adding. I can keep adding. So I have some in here. Can keep adding, and I'm forming a polymer. So this is a synthesis reaction. So if it's a carbohydrate, can be glucose, galactose, glucose, galactose, okay? Or if it's DNA or RNA, will be the nucleotide bases, adenine, thymine, cytosine, guanine. If it's RNA, you will have uracil. But basically, this is a polymer. The individual units are the monomers. So then you start breaking them, that's catabolic reaction. So now for lipids, lipids are chains of carbons with hydrogens attached to them. And lipids include fats, oils, and steroids. And then for the function of DNA and RNA, which are nucleic acids, will be to store and transmit genetic information. So here we have examples of carbohydrates, pentoses and hexoses. Pentoses have 5 carbons. In this case, 1, 2, 3, 4, 5. This is the pentose of DNA, deoxyribose, and this is a formula, C5H10O4. Then for ribose, which is the pentose of RNA, you have 1, 2, 3, 4, 5 carbons. So you have C5, you have 10 hydrogens, and you have 5 oxygens. So the difference between this deoxyribose and this ribose is the lack of this oxygen attached to this carbon in here. That's why it's called deoxy. D without oxy, without oxygen. So these are 5 carbon sugars. And then 6 carbon sugars, we have same formula, C6H12O6 for each of these. And this is the structure of glucose made out of these 6 carbons, 12 hydrogens, and 6 oxygens. And fructose, it takes a different shape with the same formula. So this is an isomer of glucose. And then galactose, it's also an isomer of glucose because it has the same formula, but it has a different shape. So it's very similar to glucose, more similar than fructose to glucose. But look at the orientation of this hydroxyl group, or this OH. So while glucose have it in this position, galactose have it in this position. And then the same in this area. So hydroxyl or OH stays here, and this hydrogen's here. An isomer has the same chemical formula, but the structure, physical structure, is different. It's like sometimes you can have the same amount and same type of ingredients to make a cake, but with this, you can also make muffins, for instance. You can use 6 eggs, 2 pounds of flour, 300 grams of sugar, and then a stick of butter. And out of that, you can make a cake, but also, and of course, you use milk or water. But in this case, you can also make muffins. And it's the same. So that's an isomer. And then those monomers, you can join them. Like if you combine glucose, which is a monomer, it's a monosaccharide, with fructose, you form sugar, which is sucrose. And this is a synthesis or dehydration reaction because you remove this hydroxyl from here and this hydrogen from here, and you join them. So that's sucrose. It's a disaccharide. Another example of a synthesis reaction is the formation of glycogen. Glycogen is made out of monomers of glucose. Glycogen is the storage form of glucose. We have it in our muscle cells, skeletal muscle, cardiac muscle cells, and also we have it in the liver. Whenever you're fasting, glucose is going to be released from your liver. because we have glycogen. Okay. So these molecules can be broken down to this glycogen in glycogenolysis, and then you will have glucose available. Now we have different types of lipids. We have saturated and unsaturated. So in this case, this is palmitic acid. It's a long polymer of carbons with a lot of hydrogens. And this one doesn't have a double covalent bond. So this structure is very rigid, so it's saturated. Whenever you have a double covalent bond, that creates a little bit of flexibility within the structure. In this case, this is oleic acid, which has a double covalent bond in here between these two carbons, and that leads to a little bit of flexibility. So this molecule, it is monounsaturated. Now, if you have more than one double covalent bond, like in this case linolenic acid, you will have more flexibility in the structure, and this is called polyunsaturated. Saturated fats are not good for the health. It can clog your arteries. Now, triglycerides, it's a different type of lipid, and it's very common in our body. That's the main form of storage of energy in our body, triglycerides. So in triglycerides, we have an alcohol molecule, a glycerol, which is made out of these three carbons and these hydroxyl groups in here, hydrogens attached to the sides, and then it's made out of three fatty acids. So we have one glycerol and three fatty acids. So this is the chemical structure of the fatty acids. So when you remove these hydroxyl groups from these fatty acids, and then this hydrogen from this glycerol, and then you release water because you form water. You form three molecules of water. This is a dehydration synthesis, removal of water, dehydration. So you form a triglyceride made out of glycerol and three fatty acids. Another type of lipid that is very common in our body are the phospholipids. Phospholipids have a polar part, which is the phosphate head, which is hydrophilic. It can interact with water because it has a negative charge. And then it has a hydrophobic portion, which is made by a tail of fatty acids, two fatty acids. And these fatty acids basically phase away the water when they are forming a membrane in cells. So then in between these fatty acids and this phosphate group, we have here a glycerol molecule, and then we have a nitrogen containing group in here. So that's the lipids, the phospholipids. Now we have other type of lipids that are called esteroids. These esteroids, they have these rings. They have 4 rings, and then these steroids that we have in our body are derivatives from cholesterol. And cholesterol itself, of course, is an esteroid. That's the chemical classification of this lipid. We have cholesterol in our body, but we have steroids that are derived from cholesterol, like we have testosterone, estrogens, progesterone, that pretty much they are similar in this chemical formula, or structure, but they have different side groups. Now, proteins are made out of monomers that we call amino acids. And amino acids, they have a central carbon, which is this. They have a hydrogen attached to this central carbon. They have an R group that varies depending on the amino acid. And they have an amino group, or NH2, and then they have this carboxyl group. So this is the core. Everything that it changes is just this. The most simple amino acid that we know is glycine. It has a hydrogen. And then we have here tryptophan, which is a non-polar amino acid. And then here we have cysteine. It's all this, of course, but I'm pointing to the difference here in the R group. So this is cysteine. This is a polar amino acid. It has this sulfahydryl group here. So the monomers of proteins, which are amino acids, can be joined together through peptide bond formation. So in here we have alanine, and then here we have glycine. So you remove water to join them. So you remove this hydroxyl group from this carboxyl group, and this hydrogen from this amino group, from glycine, and then you form a dipeptide through a peptide bond. So that's a synthesis reaction. And then you can break down the bonds between these two by adding water. That's called hydrolysis. So then you can reverse this. Now, these proteins, as they form a primary structure. So the primary structure will be represented by what type of amino acids you have in the chain. So this is a polypeptide. So here is one reading of this area. So if you increase it, so you or you magnify, you will be able to see that you have here glycine, serine, and cysteine. So the sequence of amino acids, it is called the primary structure. It's like letters of the alphabet. CAT. That's the primary structure. And then the secondary structure. So this primary structure can fold and can form two types of structures. It can form alpha helices, which are these, or beta pleats. And these are made by hydrogen bonds between the amino acids that are able to form hydrogen bonds. So you can form in secondary structure alpha helices or beta pleats. So it's foldings or coil structures that you can form. And they are stabilized by these hydrogen bonds. And these folds, you can fold them some more, and then you can form three-dimensional structures that we know as tertiary. Like some proteins can form like little spheres that we call globular proteins, or some, they can form filaments, like this fibrous protein. Collagen, it is formed by helices. It's a triple helix. We have some proteins in our blood, like albumin, that's a globular protein. It can carry drugs, or it can carry molecules. And these are as well stabilized by hydrogen bonds, but also by ionic bonds or hydrophobic interactions. So proteins, they need to have this tertiary structure in order to function. Losing this is called denaturing. So if you denature the proteins, they won't function. Now, we have a quaternary structure in some proteins, like in the case of hemoglobin, or in the case of collagen, in which you have several subunits of this tertiary structure, and then you form these shapes. Any questions so far? Okay, so for components of the nucleic acids. So in the nucleic acids, you have the unit that is called a nucleotide. In a nucleotide, you have a nitrogenous base, which will be this, a pentose sugar, and then a phosphate group. So the phosphate group and the nitrogenous bases are attached to this pentose sugar. So in the case of these different types of nucleic acids, you have purines, which are adenine and guanine, and pyrimidines that are cytosine, thymine, and uracil. So adenine and guanine has a double ring. So these have the shorter name, purines, but they have the larger structure. In the case of pyrimidines, they have the shorter structure, which is just one single ring, but they have the longer name. So an example of one of these structures. So we have ATP, adenosine triphosphate. So adenosine triphosphate has an adenine, which is this. It has this ribose sugar, and then it has 3 phosphate groups. Now, depending on how many phosphate groups you have attached to this adenine and this ribose is how you will call the molecule. Like if you have only one phosphate group, you will call it adenosine monophosphate or AMP. If you have 2, you call it adenosine diphosphate or ADP. And of course, if you have 3, it's ATP. So now, this is what happens with the use of ATP. So ATP has these phosphate groups that has a lot of energy, these bonds between these phosphate groups. So when you start breaking down the bonds, you start breaking down the bonds with the first or the terminal phosphate group, the terminal phosphate group. So you break it down in here and then you release energy. You break down this second bond in here and then you release more energy. And then as you break down these bonds, then you form from ATP first, you form ADP, and then AMP. Now, you have this core, which is ADP. You can keep adding phosphate groups, and then you can make again an ATP molecule. So this is from here to here is a synthesis reaction, and from here to here is a decomposition reaction. Now, in chemical reactions, when you release energy, this is called hexergonic chemical reaction. But in order to restore back any of these with either AMP or ADP into ATP, you have to add energy. In this case, this will be an endergonic reaction. So it's an investment. You want to generate energy, you have to spend energy. And here's the structure of DNA. So these nucleotides, they will form 2 strands. And these 2 strands will be linked to each other in the middle by hydrogen bonds. Depending on the base pairing is how many hydrogen bonds you will have. Guanines and cytosines, they have a triple hydrogen bond. 1, 2, 3. And adenines and thymines, they have 2 hydrogen bonds. And guanines always pairs with cytosine and adenine with thymine. And then these nitrogenous bases will be in the middle, and then you have the sugar phosphate backbone externally. So, if you have a DNA that has high content of G's and C's, and one that it has high content of A and T's, which one will be much easier to separate? Like to separate the two strands, one that has high content of G's and C's, or one that has high contents of A's and T's? The one that is easier to separate will be the one with a lot of A's and T's, because you have less hydrogen bonds. This one has 3 hydrogen bonds, so that makes it harder to separate. Okay, so preview of the next chapter. So we're going to learn the structure of the cells, the functions and processes like transport, communication, division, and we're going to see principles again of the chemistry level applied at the cellular level. Test your knowledge questions. Which type of bond involves an equal sharing of electrons between the atoms? Polar covalent, ionic, nonpolar covalent, or hydrogen bonds? A, B, C, or D? Unequal sharing of electrons. A. Polar covalent. Ionic is donating or accepting. Nonpolar is equal sharing. And then hydrogen bonds, there is no sharing of electrons, right? Question number 2, what does molarity measure? Number of particles per kilogram, number of moles of solid per liter of solution, number of atoms in an element, or number of electrons in a balanced shell? True or false? Oxygen gas is a molecule but not a compound, while water is both a molecule and a compound. Why is true? What is the difference between a molecule and a compound? Molecules have what that the compounds do not have? They have different properties, and then molecules are made out of the same atom. Now, ionic bonds form by transfer of electrons between atoms, creating charged ions that attract each other. True or false? Normal blood osmolarity ranges between 275 and 295 milliosmoles per liter, which helps regulate water balance and maintain homeostasis in the body. True or false? True. And then describe the 4 levels of protein structure. It will be primary, secondary, tertiary, and quaternary. Are there any hydrogen bonds in primary? Which bonds there are only? The peptide. And then these secondary can form with hydrogen bonds, while these ones can form with hydrogen bonds, hydrophobic interactions, and ionic bonds. And then this one is made by several tertiary, so subunits.